Welcome back to the deep dive. This season has been a whirlwind, hasn't it? We've really gone deep into the world of pharmaceutical development and all the systems that make sure medicines are safe and effective.
It's been quite a journey. So much is changing in this field, especially with all the new technology and the huge emphasis on data.
Absolutely. And today, we're stepping back a bit, kind of like taking a panoramic view of the landscape after exploring specific areas.
Exactly. We're going to connect some of those dots, pull out the really key analytical and quality control themes that we've talked about all season. And remember, these concepts aren't just theoretical, they have a real impact on every stage of drug development, from the lab to the manufacturing plant to the patient.
Right. And the exciting thing is, as we've seen, these stages are becoming more and more interconnected. New technologies are being integrated everywhere, and data is driving so many decisions. So today is like a cheat sheet, a way to grasp the big picture quickly.
I like that, a cheat sheet. So ready to dive back in.
Lead the way.
Right.
Where do we even begin to synthesize all of this?
Well, we always have to start with the fundamentals, right? The foundational principles of drug development and quality, the stuff that guides R&D and those very first steps in a drug's journey.
Makes sense. And speaking of fundamentals, we've talked a lot about Tuley and Kiraga's work, especially when it comes to physicochemical properties. Might sound kind of technical, but it's so crucial. Why do those characteristics matter so much right from the get-go?
Well, think about it. A drug is basically a chemical, and how that chemical interacts with the human body depends a lot on its inherent properties. Tuley and Kiraga really emphasize how things like solubility, stability, how a drug interacts with other substances. Those characteristics determine how well it gets absorbed, how quickly it breaks down, basically its whole journey once you swallow that pill.
So it's not just about finding a molecule that works in the lab. It also has to work in the real world, in a person's body.
Exactly. And if a drug can't be absorbed properly, it doesn't matter how good it looks on paper, it's not going to reach its target in the body, and it won't be effective. And remember, these properties also affect things like dosing, how often you have to take the medication, and even if you can take it with food or not, it all ties together.
Right. It's like a chain reaction. Those early properties have a ripple effect throughout the entire development process.
Yeah.
And that takes us to preclinical screening, which Teicher and Andrews discussed in detail. This is where we start really testing those potential drug candidates. What's the main goal at this point?
So, preclinical screening is all about figuring out which of these potential drugs are worth investigating further. Teicher and Andrews talk about how researchers use a mix of lab tests, what we call in vitro, and animal studies in vivo, to really get a sense of whether the drug is doing what it's supposed to and, just as importantly, if it's safe. This is where those early toxicity signals can show up.
And we've got to mention the ethical considerations here. Teicher and Andrews really highlighted the unique challenges of phase I oncology trials.
That's a really important point. When we're talking about testing new cancer drugs in phase A, often these are patients who have very advanced disease and have already tried all the standard treatments. It's a different situation than testing a new drug in healthy volunteers. The primary focus in these early oncology trials is on safety, finding a dose that's tolerable, even if it doesn't necessarily show a huge benefit right away.
So it's about walking that tightrope, trying to find that dose where you might see some positive effects but without causing harm. But then Teicher and Andrews also point out how tricky it can be to fine-tune that safe dose as you move on to phase 2 trials with larger groups of patients. What makes that transition so complicated?
Well, think about it. Phase I trials are usually smaller, and the patients are often very carefully selected. But as you expand into phase 2, you get a much more diverse group of people. They might have different medical histories, be taking other medications, things like that. And this can really change how they respond to the new drug, how well they tolerate it.
It's like you're moving from a very controlled lab setting into the more unpredictable real world.
Exactly. Plus, in phase 2, you've got more people and often longer treatment times. So you might start to see side effects that just didn't show up in the smaller phase I study. That's why getting good data early on is so important. You need a strong foundation to build on.
It all comes back to that, doesn't it? Solid data is everything.
Yeah.
And we've also discussed those in vitro permeability assays like using CACO2 cells. Chong and Ye both talked about those. Those are lab tests, right?
Yeah.
How do they help predict what's going to happen in a living organism?
It's pretty amazing, actually. Scientists can take these cells, which are derived from human intestinal cells, and create a layer in a dish that mimics the barrier of the intestine. Then they can see how well a drug candidate can pass through that cell layer. It gives them a clue about how well the drug might be absorbed in the gut.
So it's like a mini gut in a dish.
In a way, yes. But like Chung and Yi pointed out, it's not a perfect model. The real gut is much more complex. There are all sorts of transporter proteins that can either help drugs get absorbed or actually pump them back out.
Those efflux pumps, we talked about those too.
Yes, and those aren't always captured in a simple cell culture. So these in vitro models are great for early screening, but you always have to validate the results with actual in vivo studies in animals or humans.
They give you a starting point, but you need the real-world data to confirm. Now, another critical part of early development is actually refining the drug molecule itself. This is where structure-activity relationship, or SAR, studies come in. Otto Rahman and Chaudhary and Runfelt all discussed that. What's the basic idea behind SAR?
It's all about understanding the link between a drug structure and its activity. Like, how does changing a certain chemical group on the molecule affect how well it binds to its target? How does it change its properties, like its potency or how long it lasts in the body? Techniques like NMR spectroscopy, which Atta Rahman and Chaudhary talked about, are really helpful here.
NMR. That's where you use those giant magnets to look at the structure of molecules, right?
Exactly. And by making small tweaks to the structure and then testing how those changes affect the drug's behavior, medicinal chemists can start to optimize it. They're basically fine-tuning the molecule to get the best possible properties.
It's like molecular sculpting.
Yeah.
And when you don't know the exact structure of the drug target, Runfelt talked about using things like ligand-based screening and QSIR. How do those work when you're essentially working in the dark?
Well, even if you don't know the exact structure of the target, you might know about other molecules that bind to it. Those are called ligands. So in ligand-based screening, you search for new molecules that are structurally similar to those known ligands, hoping they'll bind to the same target. QSAR, on the other hand, gets a bit more complex. It involves building statistical models that connect the chemical features of a bunch of molecules with their biological activity.
So you're looking for patterns, basically.
Right. And if you find a strong correlation, you can use that model to predict the activity of brand new, untested molecules. But as Runfelt mentioned, there are some pitfalls here. If your data is inconsistent or if you have those weird activity cliffs where similar molecules behave very differently, those models can be misleading.
So you're always looking for those exceptions, those things that don't fit the pattern. And even at this early stage, before we even start thinking about making a pill or an injection, there are quality considerations, aren't there? Back in season 2, we talked about checking physical properties during lead optimization.
Oh, absolutely. Even when chemists are just starting to tweak that lead molecule, they have to think about the practicality of turning it into an actual medicine. Is it stable? Can you easily make it into a solid form? Or is it going to be some oily mess? And what about stability? Will it break down quickly in the stomach or at room temperature? Those factors can really make or break a drug candidate.
It's like you might have the most amazing molecule in the world, but if it can't survive outside of the lab, it's useless.
Right. And it comes down to cost, too. A drug that's difficult to work with is going to be much more expensive to develop and manufacture. So even in those early stages, they're trying to balance efficacy with practicality.
It's all about finding that sweet spot.
Yep.
Okay, let's shift gears now and talk about what happens once we move into manufacturing. Dissolution testing seems to be a big deal here. We talked about that in developing solid oral dosage forms. Why is it so important for quality control?
Dissolution testing is basically a way to check how well a solid drug form, like a tablet or capsule, releases the drug into a liquid medium. It's a pretty simple concept, but it's super important for making sure that each batch of a drug is consistent.
So you want to make sure that every pill releases the drug at the same rate.
Exactly. If you change the formulation or the manufacturing process, you have to check that it hasn't messed up the drug release. And even when you're just making batch after batch of the same product, you want to be sure they're all performing the same way. It's all about ensuring that what the patient gets is what they're supposed to get.
But it's not a perfect simulation of what happens in the body, right?
Right. Dissolution testing is an in vitro test, so it's done in a controlled lab setting. It doesn't always perfectly mimic the complex environment of the human gut, unless an in vitro in vivo correlation, or IVIVC, has been established.
That's a mouthful.
It is. But basically, an IVIVC means that scientists have shown a strong link between how the drug dissolves in the lab and how it behaves in the body.
So it gives you extra confidence that your dissolution test is actually predicting real-world performance. And another crucial part of quality control is making sure that the drug is pure. No unwanted substances sneaking in. Wannerberg and Persson and Beto et al. all discussed how liquid chromatography plays a big role in this. How does that work?
Liquid chromatography, or LC, is
Yeah.
a really powerful technique for separating and identifying all the different components in a drug mixture. It's like a super fine filter that can tell you exactly what's in there, including any impurities.
And why is that so important?
Well, for one thing, you need to know what you're giving to patients. You don't want any surprises lurking in that pill. But it's also about safety. Impurities can sometimes be toxic, and there are strict regulatory limits on how much of any given impurity is allowed in a drug product. LC helps us make sure we're meeting those standards.
It's all about protecting patients. And speaking of protecting patients, we have to talk about GMP, or Good Manufacturing Practices.
Yeah.
The CFR Title 21 Part 211 document lays out all those regulations. What are the main principles behind GMP, and why are they so crucial?
GMP is all about building quality into every single step of the manufacturing process. It's not just about testing the final product, it's about having systems and procedures in place to prevent errors, contamination, and mix-ups from happening in the first place.
So it's about being proactive rather than reactive.
Exactly. And it's incredibly detailed. You have to have written procedures for everything, from how you clean the equipment to how you handle complaints. It's about creating a culture of quality throughout the entire manufacturing facility.
It sounds pretty intense. But I guess when you're dealing with medicines that people rely on, you can't be too careful. And a key part of making sure those processes are reliable is process validation, which Syed Imtiaz Haider talked about. What's involved in that?
Process validation is all about proving that your manufacturing process consistently produces a high-quality product. You're not just hoping for the best. You're collecting data, running tests, and analyzing everything to demonstrate that the process is robust and reproducible.
So it's about showing that your recipe for making the drug works perfectly every time, no matter who's doing the cooking or what the kitchen looks like.
That's a great way to put it, and it's not a one-time thing. You have to continually monitor the process and revalidate it if anything changes. It's all about that constant striving for quality.
It sounds like a never-ending quest. And we've also discussed a more forward-thinking approach to quality called Quality by Design, or QBD, came up in our Season 1 transcript. How does QBD fit into this whole manufacturing quality puzzle?
QBD is about taking a more proactive and scientific approach to quality. Instead of just testing the final product and hoping for the best, you're using your knowledge of the drug and the manufacturing process to design a system that's inherently robust and consistent.
So it's about understanding the why behind everything you do, not just the what.
Exactly. You're identifying those critical quality attributes, the things that really matter for the drug's safety and efficacy, and then you're figuring out how to control the manufacturing process to consistently deliver those attributes. It's about anticipating and preventing problems before they even arise.
It sounds very much in line with that proactive approach we were talking about. Okay, let's move on to theme 3, regulatory considerations and quality standards. We touched on the ethical issues in phase I oncology trials with Teicher and Andrews. What other regulatory aspects are really important in this world of drug development?
Well, the regulatory landscape is huge. It touches every part of the process. We've already mentioned those unique ethical considerations in early phase oncology trials, but Ravina et al. Talked about something called non-inferiority trials. Those are important when you have a new drug that's not necessarily better than the existing treatment, but it might offer other advantages like fewer side effects or a more convenient way of taking it.
So it's not about being the best, it's about being just as good, but with other perks.
Exactly. And these trials require this thing called a non-inferiority margin. It's a statistical way of defining how much worse the new treatment can be compared to the standard treatment and still be considered acceptable. It's all very carefully defined.
It sounds like statistics play a huge role in these decisions.
They do, and Ravina et al. Also talked about the importance of sticking to CGMPs, those good manufacturing practices when you're working with vendors in the clinical supply chain. You know, all those companies involved in making and distributing the drug.
So even when pharma companies are outsourcing parts of the process, they're still responsible for making sure everyone is playing by the rules.
Absolutely. Quality control doesn't stop at the company's front door. It has to extend throughout the entire network. And even though it wasn't the main focus this season, we can't forget about pharmacovigilance. That's the ongoing monitoring of drug safety once it's already on the market.
It's like the safety net that catches anything that might have been missed during clinical trial.
Exactly. You're constantly collecting data, looking for any unexpected side effects that might pop up when a drug is used by a much larger and more diverse group of people. And if serious problems arise, you might have to update the labeling or even pull the drug from the market.
It's a big responsibility. But it's all about making sure that patients are protected. And in Season 5, we learned about accelerated approval and breakthrough therapy designation. Those are ways to get promising treatments to patients faster.
That's right. Accelerated approval lets the FDA approve drugs for serious conditions based on something called a surrogate endpoint. It's basically a marker that's likely to predict a clinical benefit, even if it's not the actual outcome you're looking for. It gets the drug to patients quicker, but the company still has to do more studies to prove that it really works the way they expect it to.
So it's like a provisional approval with strings attached.
Yeah, you could think of it that way. Breakthrough therapy designation is a bit different. It's for drugs that show early signs of being significantly better than existing treatments. And it gives them a fast track through the FDA with more guidance and faster review times.
Oh, it's all about speed when you have something that looks truly groundbreaking. And finally, regulations also govern things like labeling and distribution, which are covered in CFR Title 21 Parts 202 and 212. Why are those controls so important?
Well, labeling might seem like a small thing, but it's crucial. It has to be clear, accurate, and easy to understand so doctors and patients know how to use the medication safely. And distribution controls are all about making sure the drug doesn't get damaged during shipping or storage.
You don't want that temperature-sensitive medicine baking in a hot truck for hours.
Exactly. It's about maintaining the quality of the drug from the moment it leaves the manufacturing facility until it reaches the patient. It's a long chain, but every link has to be strong.
It really shows how much effort goes into making sure that medicines are safe and effective. Okay, for our last theme, let's look ahead to the future. What are some of the exciting new technologies and data-driven approaches that are shaping drug development and quality control?
Oh, there's so much happening. We talked about how omics technologies are changing drug discovery. Things like genomics, proteomics, metabolomics. They're giving researchers a much deeper understanding of disease and how drugs work at a molecular level.
So it's about using big data to find new targets and weed out bad candidates early on.
Exactly. It's about making the whole process smarter and faster. And there's a lot of buzz around connectivity mapping, which uses gene expression patterns to group chemicals based on how they might work.
So you're looking for drugs that have similar effects, even if they're chemically different.
Exactly. It could be a game changer for drug repurposing, finding new uses for existing drugs. And then there are things like pegylation, which Maibohm discussed. That's where you attach a special molecule called PEG to a drug to improve its properties.
It's like giving the drug a molecular makeover.
Exactly. It can make the drug last longer in the body, make it more stable, even make it less likely to trigger an immune response. It's pretty amazing what they can do now.
It sounds like molecular engineering at its finest. And what about those nanoparticles for drug delivery? Begg et al. Talked about those.
Nanoparticles are tiny, tiny particles that can be used to deliver drugs in new and exciting ways. They can improve absorption, protect the drug from breaking down too quickly, even target the drug to specific cells or tissues.
It's like those tiny packages you see in sci-fi movies, delivering their payload right where it's needed. And finally, we talked about how data is being used to improve the monitoring of clinical trials. Vinet et al. And Timmermans et al. Touched on that.
This is where big data and machine learning come in. By constantly analyzing the data coming from trials, you can spot potential problems much earlier. It's like having a super smart early warning system that can alert you to anything that looks off.
So it's about being more proactive and responsive during the trial itself.
Exactly. It's all part of this move toward more data-driven decision-making in every aspect of drug development. It's exciting to see where it all leads.
It really feels like we're on the cusp of a new era. So as we wrap up this deep dive, what are the key takeaways you want our listeners to remember?
Well, firstly, we can't overstate the importance of strong analytical methods. They're the foundation of quality control from start to finish. Secondly, a proactive approach to quality, like embracing GMP and QBD, is absolutely essential. You have to build quality in, not just test for it later. And lastly, we're seeing a huge wave of new technologies and data-driven approaches that are transforming the entire pharmaceutical landscape. It's all about doing things better, faster, and safer for patients.
That's a great summary. And on that note, here's a final thought for our listeners. As drug development gets more complex and data becomes even more powerful, what role will each of us play in ensuring the quality and safety of the medicines we rely on? It's a question worth thinking about. It's not just about scientists and regulators, it's about all of us being informed and engaged. Thanks for joining us for this deep dive into the world of pharmaceutical development and quality.
Until next time.
See you there.